I'm Dr. Mallika Singh. I'm a director of cancer pharmacology at a small biotech startup called Revolution Medicines in Redwood City in California in the US. There's a particular series of cancers that carry a mutation in what is called an oncogene or a driver gene for cancer. This gene is called KRAS or KRAS. And there are several cancers, a third of lung cancers, several types of colorectal cancer, as well as pancreatic cancer. Majority of pancreatic cancers carry mutations in KRAS. And thus far, it has been very difficult to drug KRAS, the enzyme itself. Over the last 5 years, there have been many efforts to drug KRAS itself. However, most of these efforts have been unsuccessful, and so people have started looking at various enzymes that are either upstream or downstream of KRAS that can modulate or affect its activity. The most recent advances have been a series of inhibitors of another enzyme called MEK or MEK. MEK inhibitors have had some success in the clinic. Unfortunately, they haven't been as successful as predicted by animal models or by other different types of models. And so there's still a huge unmet medical need for KRAS mutant cancers. There's been a lot of exciting developments in terms of drugging KRAS mutant cancers in the last couple of years. One of which has been a series of discoveries made at Revolution Medicines. And these discoveries, starting last year, were a series of observations in which we found that an enzyme called SHP2 or SHP2 can actually control the activity of certain KRAS mutations. This has the potential, particularly in lung cancers caused by smoking. We have, along with our partners with Sanofi, are developing an inhibitor of this enzyme that actually locks it into an inactive conformation. And by virtue of that, the KRAS enzyme, particularly KRAS G12C or KRAS G12A, is unable to catalyze and be active, and hence shuts down and stops cells from growing, cancer cells from growing. We are currently about to dose our first patients with a drug candidate that we're very excited about for a SHP2 inhibitor. The future for KRAS mutant cancers currently is really interesting. There are actually a series of inhibitors for KRAS, certain KRAS mutations themselves, that will be competing for this patient space along with our SHP2 inhibitor. There are at least 2 other companies that are also putting SHP2 inhibitors into the clinic. So the future looks more exciting than it ever has in my research lifetime. I think cancer is a really, really difficult disease to treat, particularly certain types of cancers. I think it's really rewarding to do cancer research, and someday those of us that are really lucky to affect a drug that goes into patients in our lifetimes. Cancer research today is very diverse and is going through an amazing explosion. Part of that is reflected in this NGBT conference, where you can see the number of tools available to cancer researchers, and the technologies that are available today are even more advanced than, say, 2 years ago. So there's been an explosion and a convergence of both technology as well as our understanding of the biology of various different types of cancers that is really creating a lot of excitement, and a lot of the pace of advances and the pace of new drugs has been steadily increasing. So it's a really exciting time to be a cancer researcher. The role of genomics is invaluable. I cannot state how important it is to understand how every patient has a different genome in terms of their cancer. And obviously the dream of any cancer researcher and the dream of the field is that one day you'll be able to walk into the clinic if you're diagnosed with cancer, that you will be able to get a personalized treatment plan based on the cancer genome that you have been unfortunately affected with.